The overall goal of the following experiment is to enable repeated measurements of the hydraulic conductivity or water permeability of the walls of venular microvessels in the rat mesentery under controlled experimental conditions. This is achieved by cannulating a venular microvessel to enable the oncotic and hydrostatic pressures in the lumen to be set and to allow the chemical composition of the perfusate and superfusate to be precisely controlled. Next, the vessel is occluded, and red cells suspended in the perfusate, which act as flow markers, can be observed moving in the lumen as indicators of transvascular fluid exchange. Then, the position and velocity of the marker red cells are recorded, either towards the site of occlusion or away from the occlusion site. Finally, occlusion is released and repeated for additional measurements, or the vessel is recannulated and measurements are repeated under changed conditions. Ultimately, paired measurements of hydraulic conductivity under control and test conditions can be made to enable the most direct investigation of the factors regulating microvessel permeability.
An advantage of the modified Landis technique to investigate the regulation of capillary permeability is the microperfusion step. Microperfusion allows the permeability of the microvessel wall to be measured under conditions where the perfusate composition, the surface area for exchange, and the microvessel pressure are all directly controlled.
This method can address questions regarding cellular and molecular mechanisms of